Welcome, everyone, to our journey into the microscopic world. Today, we are exploring locomotion and reproduction in Protista. Imagine being a single cell in a vast ocean. You have no arms, no legs, and no complex organs. Yet you must hunt for food, escape predators, and ensure your species survives. How do you do it? In this lecture, we will uncover the brilliant biological machinery that allows these tiny organisms to swim, crawl, and multiply at astonishing rates. Let us dive in and discover how life thrives at the microscopic scale. Now that we understand what protists are, let us look at the physical challenges they face. Imagine trying to move around your house without using any arms or legs. It sounds impossible, right? Protists face this exact challenge every single day in their watery environments. Because they are just a single microscopic cell, they completely lack the complex tissues, muscles, and organs that larger animals use to move. Instead, they must rely entirely on highly specialized subcellular structures. These microscopic motors and shape-shifting abilities are essential for them to hunt, escape danger, and find mates. Building on that idea of subcellular structures, let us examine the specific tools protists use to get around. There are three primary modes of locomotion. First, we have amoeboid movement, which uses pseudopodia, literally translating to false feet. The cell stretches its body out and pulls itself forward. Second is flagellar movement, where the protist uses a long, whip-like tail called a flagellum to propel itself like a motorboat. Finally, we have ciliary movement, which relies on thousands of tiny hair-like structures called cilia, beating together like oars on a rowboat. Each method involves complex biochemical interactions and specialized motor proteins. So we have seen how they move, but why does this microscopic movement matter to us? Without locomotion, protists would be entirely at the mercy of water currents. They would be unable to actively seek out nutrients or avoid toxic environments, leading to population collapse. In the real world, their movement is absolutely crucial for the health of entire ecosystems, as they form the foundational base of the aquatic food web. Furthermore, understanding these microscopic movement mechanisms helps modern scientists and engineers design tiny medical nanobots, mimicking the way a flagellum or cilium pushes through fluid to deliver drugs directly to human cells. Now that we understand the biological importance of locomotion, let us look at how we can actually measure its efficiency. While biology focuses on the physical structures of the cell, physics helps us understand the final result of that movement. We can calculate the velocity of a moving protist using the basic kinematic formula for speed, v equals d divided by t. In this equation, v represents the velocity, or speed of the protist, usually measured in micrometers per second. The symbol d stands for the total distance traveled, and t represents the time it took. If the distance increases while the time stays the same, the velocity increases, meaning the protist is moving faster. Before we move on, let us see this formula in action with a concrete example. A biology student observes a paramecium swimming across a microscope slide. We're given that it covers a total distance of 800 micrometers in exactly 4 seconds. We need to find its swimming speed. We start by identifying our given values. D is 800 and T is 4. We substitute these into our velocity formula, dividing 800 by 4. This gives us a final answer of 200 micrometers per second. This tells us that the paramecium is quite a fast swimmer in the microscopic world, using its beating cilia to rapidly propel itself toward food. Having calculated how fast these organisms can move, let us now look at exactly what these movement structures look like. On the screen, you can see a clear comparison of the three modes of locomotion we discussed. On the far left, notice the amoeba extending its pseudopodia, or false feet, to ooze forward. In the middle, we see a protist using a single long flagellum, whipping it like a propeller to drive itself through the water. Finally, on the right, observe the paramecium. Its entire body is covered in thousands of tiny cilia, which beat in a synchronized rowing motion. This diagram perfectly captures the incredible cellular machinery these master navigators use to survive. Now that we understand how they move, we must look at how they ensure their species survives through reproduction. Think about how a single successful bakery might open a second branch, and then those two open two more, quickly creating a massive chain. Protists do something very similar, but they do it by literally splitting their own bodies in half. Because they are single-celled, they do not have complex reproductive organs. Instead, they duplicate their genetic material and divide their entire being. This incredible ability allows a single microscopic cell to become a massive, thriving population overnight. In real life, this rapid multiplication can lead to massive algal blooms or allow parasites, like the one that causes malaria, to establish severe infections. To understand how they achieve such rapid growth, let us examine their specific reproductive strategies. Protists primarily rely on asexual reproduction. The most common process is called binary fission, where a single parent cell divides to form two identical daughter cells. It is highly efficient. However, nature is unpredictable. Under stressful environmental conditions, many protists can switch strategies and engage in sexual reproduction, specifically a process called conjugation. During conjugation, two individuals temporarily fuse together to exchange genetic material. This genetic mixing is crucial because it creates vital variation, giving the offspring a much better chance of surviving changing or hostile environments. This rapid asexual division we just discussed leads us to an important mathematical concept. We can mathematically model the explosive asexual reproduction of protists using the formula for exponential population growth. The formula is n sub t equals n sub 0 times 2 to the power of n. Let us break down what each symbol means. N sub t represents the final number of protists in the population after a certain time. N sub 0 is the initial number of protists we started with. The base number 2 represents the biological fact that each cell splits into exactly 2 cells during binary fission. Finally, the exponent n represents the number of generations. As n increases, the final population grows incredibly fast. Now that we have our exponential growth formula, let us apply it to a real biological scenario. A biologist places a single amoeba in a petri dish. We know it undergoes binary fission exactly once every 24 hours. We want to find out how many amoebae will be there after exactly 5 days. First, we identify our given values. Our initial population, n sub 0, is 1. Since it divides once a day for 5 days, the number of generations, n, is 5. We plug these into our equation, giving us 1 times 2 raised to the 5th power. Calculating 2 to the 5th power gives us a final answer of 32 amoebae. This perfectly highlights how effectively protists can colonize a new environment from just a single cell. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that despite lacking complex organs, protists are master navigators. They use specialized structures like pseudopodia, flagella, and cilia to move, which is absolutely essential for their survival and the health of global aquatic ecosystems. Second, we saw the sheer mathematical power of their reproduction. Through binary fission, they can achieve explosive exponential growth, dominating an environment in days. Finally, their ability to switch to sexual reproduction via conjugation ensures they have the genetic diversity needed to survive tough times. You now have a solid understanding of how these incredible microscopic organisms move and multiply.